Welcome, everybody. I'm just going to say just a couple of things before we get started. First of all, thank you to everybody for being here on a Friday afternoon. That goes for our students, faculty, and also friends and family. For those who don't know me, my name is Alex Rice. I am a professor in the math department, and I'm also the director of the honors program here at Millsaps. And this is my 7th year at Millsaps, but I've taught at a number of schools over the last, 16 years now. And, big state schools, big private research universities, other small liberal arts colleges. But in all the places that I've been, the Millsaps Honors Program is really the best approximation that I've seen targeted at undergraduate students. It's the best approximation of the experience that you get doing research in a graduate program, whether that's like a master's thesis or a PhD thesis. And so it's something that I'm really proud to be a part of. This is, I think, my 3rd honors conference as director, but I was on the honors committee before that. And, this is a very long, rigorous process for the students. They're going to come up here and talk to you for between 15 and 20 minutes. But this is the culmination of a journey that for them started in September of 2022. So they attended an orientation meeting with me to get information about the program in September of 2022. And all of the time in between has led up to them trying to distill all that down into 20 minutes for you guys. That's a very difficult thing for anybody to do, but for them to be doing this as undergraduate students is really commendable. So I think you'll all be impressed with what our students have done. And I think that's enough of my voice. I will be the moderator for today's session, so when our presenters finish, we'll give them a round of applause, and then I'll come up and ask if there are any questions. And then after questions are done, I'll ask us to thank the speaker again and give them a second round of applause. And there's no hard start and stop time for each presentation, but the speakers have been asked to try to make the presentation and question period be within 20 minutes. And then we'll usually just take a minute or two to get into the next presentation. I will say the second talk today, Keegan, will be via Zoom. He's in Canada right now, I believe, but we've done a test run for the tech on that, so that should hopefully go very smoothly. But to get out of the way, I will present our first speaker. She's doing her honors thesis in studio art. The title is Computer Assisted and Artist Realizations Working with Technology. So please go ahead and give a round of applause for Mary Noble Howard. And this works too.
Thank you. Cool. I'm assuming I need to stand in front of this to make sure that y'all can hear me, but I'm pretty loud, so if I move, it probably won't be an issue. Hey, I'm Mary Noble, and like Alex said, my project is Computer Assisted and Artist Realization Working with Technology. That artist is me, so surprise, surprise. And so I wanted to just kind of throw y'all in. So at the very start of it, there was chaos. And it started that way, it kept going that way, and you'll hear a little bit about that as I continue to talk, but it was a stressful experience, like I'm sure all the other honors projects were. But through this whole process, it wouldn't be doing me any justice if I didn't give glory to God at the beginning, because honestly, His strength got me through this, not my own, and over and over and over again, this Bible verse, my favorite Bible verse, kept going over in my head because my strength failed me, y'all, so many times. But this Bible verse says, He said to me, my grace is sufficient for you, for my power is made perfect in weakness. Therefore, I will boast all the more gladly of Christ's power so it may rest on me. And boy, did it rest on me, because she wanted to throw in the towel. So at the beginning of my honors research project, I realized that I wanted to look at how I make things and decide if that was the right way to be making things. So traditionally, I'm more of a sculptural artist. I like to be intimate with my materials. I like to be hands-on. I like to be messy. I like the chaos. like you saw at the beginning. I like the chaos. I realized that the first thing I needed to do was realize if I was keeping technology out of my artistic practice because it didn't belong there or because I didn't know how to use it properly. So first I wanted to establish my comfort zone. This is it. So this is our sculpture studio. If you haven't been in our building yet, highly recommend, go look at it. But this is where I tend to enjoy making. I like to be very messy. I like to use all the power tools. I want to be hands-on with the wood and the resin and the concrete and everything in between. And then I realized that for this project, I needed to trade it in for this environment. So this is our computer lab. And I thought that I was going to have to trade in all the things I love about making in order to make in this way instead. So trading in for these new programs, which I had no experience with prior. So that was the catch to this project, is that I didn't know how to use Blender. I didn't know how to use a CNC machine, which is my partner in crime for this project. So this is what we have in our building. It is a CNC machine, so a CNC machine basically cuts wood for you. It can do a lot of different things, and you'll have to ask our professors who are way more skilled in that area than me. But I used it strictly for cutting plywood. It can do a lot of fantastic things, but I wanted to narrow down my research into one material and one type of way. So I used 4 by 48 by 48 inch pieces of plywood to cut on the CNC with my project. And as I started to look at how I wanted to make things, I started looking at artists, obviously, for inspiration of what I wanted to make and how they were doing things. So Tadao Ando is my favorite artist. He's an architect. And Zaha Hadid, the late Zaha Hadid, is a fantastic architect in the world as well. And I sadly have not gotten to meet either of those two, but I did meet Ania Kwayamaga, and I got to have a studio visit with her after the Mid-South Sculpture Alliance conference about 2 years ago now, which is crazy. She gave me a lot of advice, a lot of hard advice about how I make and what I make. And so going forward, in our studio visit, I realized that while she was questioning how I made things and what I made, I was questioning her validity as an artist as well, because she didn't have her hands on what she was making like I did to mine. She was designing things on programs, and she was sending it off to people to cut the steel. Her husband was polishing it. She was hiring people to install it and to light the pieces. And to me, I was very offended by that as an artist, because I feel like to be the sole creator, the only person who can claim a piece of art, I thought that I needed to be the one who's doing all of the work by myself. So I was very questionable of her practice, and that kind of pushed me forward in this research. Then I started to learn about artists like Gareth Neal and Eleanor Lakeland, who are working very uniquely with CNC machines and with materiality, which in my practice I take very seriously. I personify everything. I'll personify the CNC machine as my partner in crime. I'll personify my materials as something I'm working with, not using. We're in it together. So that is definitely a key component of what I make. And so these artists started to show me that you can use a CNC machine and still be the sole artist on a piece. So I first dove straight in with my project is making chairs. So I made 3 chairs throughout this process, and I wanted to do chairs because I wanted to literally and metaphorically take a seat at these programs and these softwares that I knew nothing about. I wanted to pull up a chair and be completely vulnerable to what they're going to teach me and to all of the weaknesses that are going to come through me through this process, which were a lot. And so the egg chair, however you want to reference it, it definitely was the first and most difficult piece. So this is it. This is where it started. I went first in with the program. So this is Blender. It's a 3D modeling software that I had never used before, and frankly, I don't really want to use again. But it showed me immediately how easily these programs can show you what a physical object can look like in digital space before you put in the money, time, and effort to make it in physical form. Physical form. And so I quickly was like, maybe these are kind of cool, because I'm not a great drawer. I can probably make you anything you want to build pretty easily after some educational research, but these made it really easy for me to see how quickly these programs can change from one way of it being designed to another. So budget quickly became an issue within this project. Surprise, surprise, that is a problem. This chair by itself, the way I wanted to plan it, like this, would have cost about $2,000 to make out of plywood. And so I quickly had to change it to a flat pack design, so how these shapes slide together easily, they fit together without any kind of like screws, glue, etc. They just are kind of like a puzzle. And so this is what it looked like. So the image on your left is what the cutouts looked like on the program before they were sent to the machine. But then on your right, you can see it's exactly as it was depicted, size, shape, everything in between. The cutouts all fit together. There were some issues, but that's the learning process. And as you can see, like I said, it's exactly as it depicted, which was a huge relief to me because I didn't want to have to go back into these programs. And so as the skeleton of it is coming together, pretty much exactly as I need it to, I still wanted it to be a solid object. I still wanted it to be not see-through, not have holes in it. I wanted it to be like just a nice, solid object. And so I decided to do a papier-mâché type style on the exterior of the egg with muslin and cement. And so that is where I started to see that me as the artist can integrate different aspects of what I like to do instead of having to figure out how the cuts need to be, what size they need to be, how they fit together. I realized that I can go in instead and treat it as more of a surface treatment so that the integrity of the chair is not compromised by my issues of, engineering education, but quickly I realized that I can spend my time in different ways. So this is what it started to look like. As you can see, the skeleton on the inside with the surface treatment on the outside. And this is us moving it. It is very heavy, but it quickly became, thought I had another image there. I'll show you later. But this is the final chair, and so I quickly realized that this piece was something for me that changed the way I approached the pieces going forward. So my next chair is the pine cone chair, and unlike how the egg chair started, this chart started with a sketch, and so that's how I normally start my pieces. I decided with this project to really focus on how the technology can work for me instead of forcing myself to go through Blender, VCarve, Slicer for Fusion 360, Illustrator, all of these different programs that I used for the egg chair. I wanted to limit them down so that I was only doing what I needed to do to make what I wanted. So I went directly into Adobe Illustrator for this chair versus going through all the others. And this is the only cuts that I did for the pine cone chair. And I realized that it was a lot simpler, a lot less complicated. It seemed to be what was working for me better than Blender. We aren't friends. And so, like I said earlier with Eleanor Lakeland, who's an artist who I took inspiration from, she works a lot with materiality, and she's very intentional about what she chooses. And I'm born and bred Mississippian through and through, so I decided to use a cypress beam from Mississippi in order to put my heritage back into my pieces and to really focus on integrating different aspects of materials into each chair that I was doing. And so that's what this was. This is me assembling it. So with the other one, it was me throwing cement and heavy pieces of wet fabric on top of a skeleton, but this one was more stacking and hammering. As you can see, it's coming together. This is me kind of deciding on the weight distribution, and as you probably noticed, it's different than the sketch, but as an artist, you have to kind of go forward with, issues and things like that, like weight distribution. So this is how it ended up looking, and so it's a lot different than the sketch, but I was really happy with it because I was able to work with these technologies in a way that made me feel comfortable, made me feel like as an artist I had more control. So the last chair that I did is titled The Rocking Chair, and it's an open source file. And so with this piece, as I was slowly removing more and more of the technology, I decided to remove myself from this piece as well. So an open source file is basically one that's online. You can find it. It's really easy. If you have access to a CNC machine, you can go in and use that file to make the object yourself. So that's what I did, and I realized that I am the problem when working with the CNC machine. All of the issues that I had going into it, all of the problems that came through my creation process with the egg and the pine cone, they weren't a problem with the rocking chair because the cuts I nested, so put them onto smaller pieces of plywood to save material, to save money, to save time. But really, all the cuts were ready to go. And so I wanted IKEA-like instructions. I did not get them. But really, the only problem with this chair specifically was figuring out how it was going to fit together, which quickly, me stacking it, me figuring it out, it wasn't hard. It wasn't difficult. The CNC did exactly as it was supposed to do, and it was a lot smoother process than some of the other pieces that I designed from scratch. So this is the way the chair ended up looking. On your right, that's the stock photo that I had to work with, the only image of the chair that I had to work with. And all of those shapes are totally different. So figuring out how they stack together was slightly a headache, but a lot less chaotic than I was expecting. And on the left is my chair that I finished with. So the three chairs all taught me something different. The pinecone chair taught me how I can make technology work for me. The egg chair taught me that sometimes you need all the technology, and that's great, but I'm not sure that I do. And the rocking chair taught me that I definitely am the issue when learning technology, but as I went forward, sorry guys, that's me. In the egg chair, I do fit. So as I go forward, I realized early on that it wasn't me and the CNC against each other, it was just how we could come together. So as I approached it originally, wanting to beat the CNC machine, be like, I don't need the technology, I don't want to work with it, I don't need to have it in my practice, it really wasn't that. And so I realized that at the end of this, I made a side table and that I could put how I make art into effect with these programs. So this is actually a side table with leftover beam from the pinecone chair, so the remains of that, but it's puzzle pieces in between that all fit together on a program. So if you laid them flat, they would all fit together like a puzzle. They're different sizes, they all work together. But the idea essentially here is that it stacks up, like life's full of lessons, and sometimes it doesn't all lay flat and it doesn't all work out how you want it to, but you're all, at the end of the day, gonna have a fantastic something to look at. You're gonna have a fantastic lesson that you learned, that everything builds on itself and it all works out because it all stacks up. And so I realized that my conceptual thing that I love so much about my pieces that I removed from these chairs, I didn't want to put too much meaning into them. I just wanted to work with how to work with them. I realized that I could add it back, and that technology at the end of the day is not something that I'm against because I've learned how to use it better than I used to, but I still think that my integrity with my materials is not compromised by using the technology. It just changes, and I think it's okay for relationships to change. This was my exhibition that just got taken down about a week ago, so I had a whole exhibition in the gallery. It was awesome. I was so lucky to have everybody there supporting me. And speaking of support, I have to give acknowledgments to my fantastic faculty. So Eric Charlton was my first reader, Sue Carey Drummond was my second reader, and Laura Franey is my third, who's not pictured but equally as awesome. And that was us presenting all of our personal research in Richmond, Virginia this October. So I just want to say thank y'all so much.
Does anybody have any questions for Mary Noble?
That's a great question. I don't know. We ended up having to move it with that crane, and honestly, it probably weighs like over 300 pounds, maybe. I'm trying to get confirmation from the men who actually helped me lift it. About 300 pounds, probably, minus the square that it sat on at the bottom. It's a great question.
Any other questions? Kurt?
So I don't know if this was your first introduction to this kind of new technology or if you dabbled in it, but has it changed your idea of like future projects that you might have thought were out of your realm, but now you're like, wait a minute, I can do this now?
No, that's an awesome question. So kind of working with the side table is like, it was a last minute kind of addition to this project, but I realized, cutting out curved pieces of plywood, if anybody's worked with plywood, it's really hard to cut, because it wants to split, it wants to do all kinds of things, but the CNC made it really easy to make curves and stuff like that, and that's just a small example to your question. Yes, I do think that it would change the way of thinking about bigger and grander things that I could do in the future, but I do still think that I love being intimate with my materials, so as long as I can do both, then I'm not afraid of using technology in any regard. Yes?
Which program do you find the easiest to work with?
That's a great question. I think VCarve is definitely one of the easiest, so that's the program that speaks to the CNC machine and tells it what to cut and how to cut. And for me, that was the easiest to work with. Blender was the extreme end of that. That made it extremely difficult, and I was like, we're going to keep some distance between us for healthy reasons. But I also use Adobe Illustrator, which if you have any experience with digital technologies, that one's pretty beginner-friendly, and I think that, like the Pinecone chair is what I used for that program. Is what I used for the Pinecone chair, and that was really simple for me to figure out.
The program that you said was linked to the CNC, does it have basic stuff in it, or is it?
Yes, it took a while to understand kind of how things worked and like figuring out like designing the cut paths is really the terminology for that, is figuring out where the CNC is going to cut so you don't actually slice one of your things in half. That was the only complicated part, and so really that, once you had that going, you could probably figure out anything else. And it has its own nesting qualities in it, so if you wanted to do that like through the program, you could do that, but I just didn't like my hand.
Got time for one more, if anybody has one more question.
Say if someone wanted to purchase the rocking chair and moving to capitalism, about what would it cost to retail that chair?
It's a great question. I think it just depends on, because I didn't design the chair, it's hard for me to place a budget on that, but material-wise, I think it was like $200, but I think time-wise, I'd probably do around $1,000 for that chair, personally.
I'm not.
Sue Carey and or Sarah have
Can I just ask a quick
questions.
question? Sorry. With that issue going up, I hadn't thought about this. What do you think are the ethical quandaries of you selling that chair? Since
That's a great question. I know, I was like, I don't know.
But you paid for the design.
No, exactly. No, that's a great question. I think I don't think it's a question, because I'm in no way claiming this chair is my own creation. I don't think it's a plagiarism issue. I think it's a learning tactic for this project, but I don't know how I feel about selling it. I think it would be really weird to sell somebody else's design, but I did fabricate it. I don't know, it'd be definitely something to pray about and talk to my therapist about.
Sarah, did you have something else?
Yeah, mine was more of this sort of conundrum that I noticed, and in that you of Zaha Hadid, and her
Yes.
design would not be possible without technology.
Absolutely, yes.
And then you're so like anti-technology with those projects.
No, absolutely. And I think that is definitely a huge part of it because I realized that I admire so many things that people are doing with technology. So I think that was definitely something that stemmed me and pushed me further into this project of like, okay, can't be a hypocrite. Like, let's figure this out for ourselves. And so I definitely recognize all that her and Tadawanda do. And I think now I have more of a better understanding of how they do it and what makes them so special as more of the designer of the project, not the creator of a physical object.
Let's thank Mary Noble one more time. Just take another minute or so. Let's see. There we go.
My computer audio.
Keegan, you are live. Can you hear me?
Yes, you're a
Okay.
little quiet,
All
but
right.
I can hear you.
We're going to take just a minute or so. Why don't you go ahead and share your screen,
Yeah.
and I'll introduce you in just a minute.
Alrighty. You disabled screen sharing again.
Okay, sorry. Let me do that. All right, try it now. All right. Great. Okay. When I introduce you, I'm going to go ahead and let you say the title of the presentation.
Okay.
Well, we're right on schedule. It's 2:10. So, live from Canada, is that right?
Yep.
All right,
Canada it is.
live from Canada. Our next presentation, doing his honors project in biology, is Keegan Peschke. So let's give Keegan a round of applause before he starts.
Great, cool. I can't hear anything, so I'm going to assume I'm good to go.
Yep,
So
it's
my
all
talk-
you.
All right, great. My talk is on the effects of mutation in the FEMA and HDFR genes on biofilm formation and how they affect the symbiotic relationship between Xenarabis nematophila, which is the bacteria that you can see in this picture right here, and the nematodes Steinernema carbocapsae. So, Great. So what are we talking about now? So Steinernema carbocapsae is this nematode that you can see right here. It has a mutualistic relationship with the bacteria Xenarabis nematophila. In this mutualistic relationship, the nematode is going to provide transportation for the bacteria and protection from the environment. The bacteria, in turn, is going to provide the nematode with nutrients and the ability to reproduce, which we'll talk about more in a minute. The reason that we're particularly interested in this bacteria and the nematode is that this bacteria is unique in that it has both mutualistic and parasitic relationships. So there are insects that like to eat, say, tobacco plants, tomato plants, these ones right here. And because they eat our plants, as you can see, that are going to destroy our delicious crops, we're very interested in how they interact with each other and how that can relate to human biology, which we'll talk about more in a minute. So, a bit of a vague background of what's going on here. So if we start at the top in our bacteria nematode complex, which is this picture that you can see right here, we have a biphasic life cycle. So this life cycle starts by the bacteria and the nematode. They're in the soil. The bacteria and the nematode, they're in the soil looking around for an insect. And eventually they're going to find an insect that they can get into, and they're going to enter the insect. From there, the nematode is going to release the bacteria into the insect, and it's going to start producing virulence factors, or basically things that are going to start to kill the insect. Eventually, once the bacteria has killed the insect, we're going to have a lot of excess nutrients because we can begin to break down our host tissue. So the bacteria has provided nutrients to the nematode, and now it's going to get the ability to reproduce because as we have our little life cycle right here. And since there's all these extra host nutrients around the bacteria, and the nematode can reproduce. Once those nutrients start to go away and we start to run out of nutrients, the bacteria and the nematode are going to reassociate, or in other words, the bacteria are going to colonize the nematode, and the nematode and the bacteria are going to exit our insect cadaver and restart this cycle. So there's two spots that are very stressful in this relationship, in this life cycle. Right here, when the bacteria is first introduced to the insect, and when we reassociate with the nematode. When we first associate with the insect, it's very stressful because, like humans, insects don't want to be invaded with bacteria. There's a lot of host immune responses. It's going to try and kill the bacteria. And when they reassociate, because there's not many nutrients left, it's in a non-feeding state, and it's very stressful for the bacteria and the nematode. So this means that we have to be able to live within two hosts that are stressful. So how can we do this? The answer is biofilms. So biofilms are a response to stress. This stress can be a lot of different things: pH, lack of nutrients, host immune responses, other stuff. These biofilms are going to protect the bacteria and allow it to spread nutrients between little bacteria friends around here. So in this diagram, which isn't the best diagram, you'll see these little blue lines right here. These are what we call pili. They're what we use to attach to the surface on the, like this surface right here, this little black surface. We're going to use these pili to attach to them. This will be important in a minute, but that's how we initially attach. And once we have attached, we're going to start making this peach colored, brown colored, whatever color you want to call it stuff. It's basically just sugars and nucleic acids. So it just basically think of it as a sugary matrix is what we would call it. These biofilms are believed to play an important role in associating with the nematode or the insect, or in other words, hosts in Xenarabdis nematophila, but it hasn't been studied yet. So that's why we're doing this, because no one studied it. We don't really know. It's assumed, but we just have to find that out. Why do we care about biofilms? So biofilms are very common in human things. So here's a list of examples. The most common of which that you've all had the chance to deal with is oral biofilms. Or in other words, you know that lovely plaque stuff you get on your teeth? That's a biofilm. They can lead to chronic infections, and they can be difficult to remove. And the more that we can learn about these biofilms in other bacteria, the more that we can apply it to humans. So like I said earlier, since this is unique in that it does both mutualistic and parasitic relationships, studying it can provide useful data. So what are we actually doing? In really big words, targeted mutagenesis of putative biofilm-associated genes. Or in other words, we're mutating genes that that are associated with biofilms. So FEMA is one of the genes that I'm working with. It is the synthesis of the pili precursor. Or in other words, it's what's making those little blue things that I pointed out earlier, what initially attaches the biofilm to its surface. And the other one is HDFR, which is a negative flagella regulator, or in other words, it is what is telling the bacteria to stop making flagella. And the flagella is, think of it like a tail that you can use to swim through your solution or whatever. That's the best described for it. Then what? We've mutated the bacteria. What are we doing now? The next thing we would see is that we're going to grow biofilms. We're going to assess those biofilms. So see, are they bigger? Are they smaller? Are they distributed differently? Are they all over the test tube in the same spot? And then how are they going to colonize the nematode? So if there's less biofilm, we're going to assume there's less colonization. So when we mutate our bacteria, what we expected is that the FEMA mutant, because there's less pili, there will be less adhesion, and that it will make a bigger community and there will be more nutrients. It's kind of what we're expecting. With the HDFR mutant, it's a little bit more complicated. And so the flagella, as its role is movement, since we're not deactivating the flagella anymore, we're going to keep moving through our solution. So when we try to attach to our surface, it's hard to stick your brakes on something when you're still pushing on the gas. The flagella is also very energy costly in that it costs a lot of nutrients to build it, and it also costs a lot of energy for it to keep moving the bacteria. So we're losing nutrients. And as you remember, biofilm is a stress response where there may not be a lot of nutrients, so that can be very costly for our bacteria. So how are you going to mutate this? So basically what we're starting with here is this is a circular piece of DNA. We call it a plasmid. You can see it has something on it called CAT that codes for antibiotic resistance, and there's a SAC1 site right here. Basically what we're going to do is we're going to cut that SAC1 site. We're going to insert part of the target gene, so FEMA or HDFR right here, and then we're going to have it combine with the Xenarabidis nematophylla DNA and insert itself into the chromosome so that we have now deactivated the gene because we've stuck something in the middle of it. It can no longer copy correctly. So how are we doing that? So first thing we needed to do is PCR, which is basically a DNA copy machine. We're just adding, brain. Sorry, we're replicating the DNA. So first, to do that, we need to make primers for our DNA that's going to amplify like an internal portion of it, and that are also going to encode for our digestion enzymes recognition site, which we'll get into in a second. This fragment is what we're going to use to insert into the gene. I unfortunately neglected to edit this, so we did a couple of different methods at different temperatures to see which primer would work the best, but we ended up just using all of them. So basically, this is our proof that we have our little internal fragment of the gene. So this is part of our gene. So from there, we're going to take that plasmid, we're going to digest it, which means we're going to cut it open up that SAC1 site. And yeah, so from there, we can ligate it, which I have a nice pretty picture, which is really good to explain this. So this blue section right here that you can see, if this is our internal fragment, it has this little sticky end right here. When we cut open our plasmid, we also got this same sticky end on the other side. So we can ligate them together, and now we have our plasmid with our internal gene fragment on it, which you can see here. So now this is what we have now. Our plasmid's ready. We can get it. We are now able to recombine it with our Xenarabidis nematophylla. But there's a few more steps before we can actually do that, which would be transformation. So basically, what we had before is just a loose plasmid hanging out in solution. It's not currently in a cell, so it's very difficult to work with. So we want to put it in a cell. The cells that we chose is E. coli. So basically, we're having our E. coli uptake the plasmid. So now we have an E. coli cell with our plasmid in it. And from there, we can select to make sure that we have E. coli that actually uptook the cell. And then we can run, this is what a PCR looks like once you've ran a gel of it. We took the FEMA and the HDFR that actually grew. And yeah, this is how we know that it has the plasmid that we want, and it's in our E. coli. So from there, it's in E. coli. It's not in Xenarabidis nematophylla. That's not going to work for us. So how are we actually going to fix that? So we're going to grow some fresh Xenarabidis nematophylla, and we're going to mix it with those E. coli cells. And from there, we'll get a plasmid donation or a conjugation, where you can see if this is our E. coli cell right here, we're going to conjugate with the cell, and we're going to donate that plasmid or replicate it and give it a different plasmid. Oh no, the picture's on the other side. Okay, unfortunately, the picture's on the wrong spot. It's that picture from earlier. It's the plasmid. We're going to do our recombine with the DNA, and it can now irreversibly insert itself into the DNA. So we've now mutated our gene. It's deactivated. It no longer works. So how do we know that actually happened? Basically, since Xenarabidis nematophylla encodes for antibiotic resistance itself, and so does the plasmid, we can selectively grow it on different plates to make sure that they grew properly. And from there, in order to get rid of E. coli-based contaminants, we can select colonies and grow them on new plates until they're nice and pure for us. So biofilm experiments, now they're actually to that. So short and dirty is we standardized the starting conditions for all the cultures. They're dyed with crystal violet dye for 10 minutes and then rinsed a couple times. We dried them overnight and then measured them with a spectrophotometer. So results, what actually ended up happening? So FEMA, like we expected, had less biofilm compared to the wild type, which I'll show you in a moment. It had a p-value of, there wasn't a lot of trials, so the p-value is not perfect, but it's less than 0.05, which would mean that it's significant. This is what it looks like. So right here is our wild type Xenarabdus nematophila, and here is the FemA mutant version, which you can see if you look at the lines, there's a lot less, excuse me, a lot less biofilm on this one. The HDFR was interesting. So as you can see, the p-value is above 0.05. So there wasn't a significant change in biofilm formation. However, the distribution of the biofilm is kind of, instead of in a nice, concise line, it is all over the place, as you can see. So what we are thinking is happening with this is that because we didn't deactivate our flagella, it kept trying to attach to the surface, and then it didn't attach and started to try and grow the biofilm in solution until it hit like a glass wall or something like that, and was unable to do what it was supposed to do. So, unfortunately, we ran out of time to actually do all of the things that we want to do. So, in future, what we would do is introduce the bacteria to the nematode and let it colonize it and determine how that mutation changes the colonization in the nematode. If there's less biofilm, we'd expect there to be less bacteria in the nematode, is currently the hypothesis. With the insect, as you can see, we'd also expect kind of the same thing, that if the bacteria can't hide from the host immune response with its biofilm, that there should be less of them because they're dying. I also have some acknowledgements I need to make. Of course, my advisor, Dr. Hussa, who provided me lots of guidance, time, expertise, resources, space, all that fun stuff. The honors program for giving me the opportunity to do this and complete this project. My committee for offering suggestions and donating their time to help me succeed and edit that paper, because that was fun. And of course, the college for giving me space and funding and resources as well. Great.
All right. If anybody has questions for Keegan, you can ask them and I'll relay them to him through this microphone. So does anybody have any questions for Keegan? All right. Well, Laura, yeah.
So is this part of a larger research project, and what kind of role, like how did you work with Dr. Hussa to come up with the ideas? Were you coming up with some of the ideas of what to do, or mostly, kind of doing the experiment?
Yeah. So the question is this a component of a larger research project, and how did you work with Dr. Hussa to sort of come up with what you were doing with your experiments and all your methods and protocols? So what did that process look like?
So it is part of a larger project. Dr. Hussa is actually her next honor student, is doing some different genes, and if I remember correctly, is doing a Ford Fellowship where she's actually continuing like the biofilm experiments that I had to leave, and the insect would get to the nematode colonization stuff if she also has time herself. As far as working with Dr. Hussa, she provided some resources that can suggest genes that might have been good to research. So one of the two genes I picked was from a paper she recommended. The other one, I went and found my own paper. As far as protocols, a lot of this is kind of standardized protocols. So she just was like, hey, here's kind of what you should probably consider doing. There had to be a few modifications as research tends to go, but that's kind of how that went, if that answers your question.
Thanks. Any other questions for Keegan? Kurt?
The changes in the biofilm, is that better or worse for the insect? In other words, if that was altered, does it make the insect less likely to die, or not?
The question is, the changes in biofilm, are those better or worse for the insect that is infected?
So right now, that's mostly hypothetical. We haven't gotten a chance to study that yet. However, if I had to take a stab at it, if there's more biofilm, that means there's going to be more bacteria. The insect's going to die faster. If there's less bacteria, the insect might be, or sorry, less biofilm, the insect might be able to fight off the bacteria, and so it doesn't die. But it would probably, realistically, if there's any biofilm, it would probably die either way, because the nematode also tries to help, does a little bit to help kill the insect too. So ultimately, less biofilm would probably help the insect, but it'd probably still die either way.
All right. Well, let's thank Keegan one more time. All right. Thanks, Keegan. Have a good rest of your day.
Yep.
Okay. We'll just take about a minute or two before Annabelle gets started. How's it going? There we go. Okay. So this clicker should work. So you can use that, or you can. Presentation. Our next speaker did her honors thesis in psychology. Her title is Self-Perceptions and TikTok Among College Students. So please give a warm welcome to Annabelle Bailey.
All right, so as stated, I did my honors thesis on self-perceptions and TikTok among college students. So just a quick question, how many people have TikTok? Everyone, I see everyone. Dad, raise your hand, you have it, I know you have it. Yeah, everyone under 30 has it. So hopefully you can learn something from this. So why kind of research about TikTok? Current research is not up to date regarding the newer social media platforms and its impact on self-perception and body satisfaction. With so many people on that platform, over 2 billion users, it was just something interested in me, and anytime I searched up social media, TikTok, nothing really popped up. So I was like, why not? I started to kind of curate these questions about TikTok. Do users' self-perceptions alter when watching TikTok? Do users create more social comparisons? Do users' body satisfaction decrease? How does anxiety and depression contribute to self-perception and body satisfaction? And are there any gender or ethnicity differences among users on how they perceive things when they're watching videos? So the goal of the study was just to understand to the extent in which TikTok impacts self-perception and body satisfaction, as well as looking at those depression and anxiety. Does it really contribute, or is it kind of a myth?
my hypothesis was that TikTok that invites physical comparison, so anything that you're watching a person dancing, singing, doing their hair, would be associated with decrease in body satisfactions compared to those who didn't watch those physical comparisons. So participants were recruited from Millsaps College. We had 92 students. All of them received course credit. Participants ranged from 17 to 22, but with an average range about 18.87. I had many questionnaires. First, we had our demographics questionnaire. Then we had a body comparison scale, which is just rating your body part from 1 to 5. It could be a nose, eye, ear, how you feel about it personally. Then our depression scale, just questions about depression. How often in the past week do you feel lonely? Are you feeling sad today? Things like that. We had a multi-dimensional body self-relations appearance scales questionnaire, and that's just kind of looking at your body and be like, am I sexually appealing? Do I feel like I'm better than other people? Am I not as sexually appealing to other people? Then we had a physical appearance comparison scale, just when you're out and about, how are you physically comparing yourself to others? Then we had our physical state trait anxiety scale, just how anxious you feel in general. We had a social cultural attitude scale, just kind of looking at like how you feel towards your social peers. I feel pressured from my friends to look skinny. It even talked about when we were looking at social media, how you feel pressured when looking at things on social media. And an upward, downward physical comparison scale, which is how do you tend to compare your own physical attractiveness to other things. So my methods, first, our participants were just randomly assigned to one of the two conditions. We had our experimental group or our control group. So the experimental group just viewed those TikToks that had exposed them to a range of physical attributes. We just had hair, face, body, and eyes. And I have a couple of videos to kind of show you about what they watched.
And who am I? That's one secret I'll never tell. The only one. XOXO Gossip Girl.
This was just showing that we showed people with different clothing styles, different genders, different ethnicities, and then we also showed.
I'ma throw this money while you do it with no hands. Go.
My control group viewed TikToks which did not expose those physical attributes, so we really talked about different categories such as baby animals, drawings, cooking, and our travel scenery. So those looked like this.
How can you miss someone you never met? 'Cause I need you now, but I don't know you yet.
Then my favorite personally.
Hey there, what's your name? Maybe want to be my buddy?
And then I had some travel scenery as well. So my methods is, before they even watched the videos, they completed that whole packet of questionnaires. Then they watched the TikTok videos for about 5 to 6 minutes. Then after that, they're gonna repeat those state trait anxiety scales and the upwards and downwards questionnaire to just kind of see and measure their self-perception after they watch the TikToks. We're moving on to results and discussion of procedures. So ANOVAs can seem like a scary word, but it's just something, I'm comparing something to something else. So I'm taking one group and I'm just comparing it to see if there's a statistical significance. So to see if something happened. So group memberships and pastas. Group membership is just that control group and that experimental group. Pastas was just taking it before and after, and it's that state trait anxiety scale. So there was significant difference between the pasta responses before viewing videos versus after. This could highlight that participants' anxieties about physical appearances could be altered when you're watching TikTok in general. The PASTA scores for the experimental group were slightly higher, so it could imply that participants maintain their previous anxieties when watching those TikToks. And the PASTA scores for the control group decreased, and this could be because TikTok could have relaxed them, and the participants were leading to lower physical appearance anxiety. Then we have group membership in VOS. So VOS is just that upward and downward scale. The experimental group had higher social comparison rates, which is that VOS, and then the control group. This could be due to the social comparison theory, as people have a self-drive to evaluate themselves when they see another person. Due to the control group not including those people, those participants did not automatically engage in comparisons to leading to fewer social comparisons. I probably am not going to compare myself to a goat when I see a goat on the TV, so I would probably compare myself to another girl or anything like that. 3 by 2, I'm just taking 3 groups and another 2 groups, and I'm just comparing them. So gender, we had 3 genders. We had males, females, and non-binaries, and PASTAs is that state trait anxiety scale. Females had significantly higher scores in comparison to males and non-binaries. This could be due to females being just more anxious about themselves and their physical attributes and how they present themselves to society. Females' self-judgment could take precedence over positive or reassuring thoughts, leading to anxiety and being more critical to societal expectations. Females are more observant, and we can utilize visual concepts from TikToks to make comparisons when the TikTok is displaying another person. So with the social comparisons, females made more social comparisons before and after than other genders. The difference between genders could be due to other genders being more socially isolated than females. Females tend to be more socially connected and value those interactions with other people. Therefore, we're just going to engage with social comparisons more frequently than others. One way ANOVA is I'm just comparing one group. So one way is for gender. Depression was more prevalent among females. The increased depression rates could be associated with that greater uncertainty and may increase social comparisons. So the body comparison scale is the BCS, and the physical appearance comparison scale is that PAX. Females tend to have a higher engagement with body and physical appearance comparisons. They tend to have a higher body standard than other genders. Body issues are more prevalent from females as they tend to be more socially compared, upward, leading to self-critical comparisons. And males tend to be more hopeful about their body image, and it kind of seems to have less societal pressure. In the SAT Q4, there is a relationship between weight concerns and social cultural attitudes towards appearance, and so females tend to be more concerned with weight, therefore could explain this gender difference. So some additional results that I had was there's an understanding that those with high appearance anxieties are likely to have high rates of depression and increased social and physical comparisons. And this I found really interesting is that African Americans overall, their physical appearance comparisons were significantly lower, indicating they just have greater body satisfactions than Caucasians. Data is consistent with previous research and found that African Americans have fewer discrepancies related to their body dissatisfaction and more positive cognitions about their body images than other ethnicities. So what I've kind of learned from all of that is my study was able to provide insight in the relationship between social media content, individual anxieties, gender, ethnicity, depression, and the role of social comparisons in shaping how somebody thinks about themselves. There are consequences to watching those TikTok videos. Females or those with higher level depression rates should be more cautious when participating on social media. So we really should be limiting our time and what we're doing when we know that we are more prevalent to have that. And TikTok impacted users' body satisfaction and self-perception regardless of which group they were in. Further research may contribute to development of intervention strategies to promote to a healthier self-perceptions and body satisfaction when viewing curated social media content. And this is just a huge thank you. So these were all the people who helped me collect my data over the past 2 years. Julia and Asha sit in the back, and they helped me as well go through the process of writing. Dr. Groundhouse killed it. Alex killed it. Kurt killed it. They all helped me, made me believe in myself. And to my dad and my grandpa for helping me push through school and, you know, believing that I could do it every time that I called crying that I couldn't. And to Gavin for being there for me as well. And also Zeus, if you've seen him around, he's my lovely golden doodle. So these are just, let me go back.
Does anybody have any questions for Annabelle?
So you said the participants saw videos that were related to hair and all that stuff, right? Do you think the participants' demographics and the video, whoever is in that video, their demographics correlate somewhere and could skew your results?
No, because I can only show you a small skew of what I had. Those 5 to 6 minutes, if you would love to see all the videos, we purposely picked different genders, different hairstyles, making sure that everyone was represented, so it wasn't just based on one thing. So I personally don't think that they were curated.
So the popular conversation around social media and body image tends to focus on social media as one sort of gigantic category of media that might have a negative impact on one's body image. But I wonder, just theoretically, if you think that there's something unique about the TikTok interface, the way that it presents content to people, the way in which people engage with it specifically, compared to other social media platforms that makes TikTok of particular interest to the conversation.
To get into this scrolling thing. It's very reinforcing to keep scrolling, and TikTok is curated. Since I curated my own thing, but if I opened up your TikTok and my TikTok, they're 2 separate different things. They want to keep you invested. They want to try to make you stay on longer. So I feel like in terms of Instagram, and other social media platforms, it's just something about how fast-paced TikTok is that it's just, I feel like it's got a little bit of edge on other platforms.
Do you still use TikTok?
I do not use TikTok. I deleted it off my phone after I found you. Well, I do not use it anymore. I'm an avid Instagram user.
Yeah.
If you were president, would you advocate banning TikTok in the United States?
Great question.
She told me not to ask questions.
I would probably advocate it on the research that I've done because I know how beneficial it can be to step away from social media. So I would probably advocate that, yes.
Yeah, I just want to make sure that I heard you correctly. Did you say that these surveys were administered after watching a 5 to 6 minute compilation?
Yes.
Mary Frances?
What process, or was there any sort of process in how you curated the different compilations of TikToks for your groups to view?
It took months. Yeah. Sending it back. Well, because I didn't realize how biased my thought process was. I love, cows and dogs and all the things, and Groundhouse would sit there and be like, well, what if somebody doesn't like a dog? What if they hate cows? What if they hate dogging? What if they don't look like that? And it was really, I needed to understand what representation was. And so it did take a while, and I finally understood it, but we did it, so.
Did you ask
the participants, like, if they already had the app downloaded before or not, and did you see if that, like, had an effect on the results at all?
No, we did not ask that. So
Was there any positive results that you found after your control groups used TikTok?
what do we believe is positive? Pretty much it was, I wouldn't use the word positive, I'd really just say there was just a difference. It was just to be able to say that they did this, watch this video, and there was a different outcome than before.
I have a question. So I have kids, and we don't do TikTok, but you said that you are okay with Instagram. So how do you feel about that Reels kind of create the same TikTok effect?
I personally feel like Instagram and TikTok are two completely different things. The Reels on Instagram, I'm not saying that they're not different, it's just something about how quick it is on TikTok because it's a newer platform to be able to upload a video and then just keep scrolling. Like, for me, personally, Instagram, I can just shut it off. And TikTok, you'll just sit there and it's like 2 o'clock in the morning. So it could just be depending on what you prefer as a platform, but I believe that they're just two different things. And I couldn't compare the Reels on Instagram to the Reels on TikTok.
I don't have TikTok to compare it, so I just was curious.
If you want to download it and compare it and do the research on it, let me know.
All right, let's thank Annabelle again. We have 2 more presentations left. And we'll go ahead and we're right on schedule, so we'll go ahead and get started. Our next presentation is an honors thesis in geosciences. The title is Analysis of Microplastics in Fluvial Sediment of the Pearl River, Central Mississippi. So please give a warm welcome to Willie Ponte.
Good afternoon, everyone. Everyone hear me okay? Okay. Before I begin, well, first of all, my name is William Ponte. This is Analysis of Microplastics in Fluvial Sediment of the Pearl River in Central Mississippi. But before I begin, I'd like to dedicate this presentation and all the work I've done in loving memory of my grandfather. I know he has the best seat in the house, and so without further ado, let's get into it. Plastic is ingrained in our society in numerous ways. I'm willing to bet everyone in this room has come into contact with some plastics in the last hour, the last 30 seconds maybe. There are over 5,300 different polymers in existence, each with their own chemical makeup and different chemical properties. But one chemical property that makes plastic so pervasive in our society is that it's tough, right? So that makes it difficult to biodegrade in a natural environment, and this leads it to being a threat to our environment and our ecosystems. But what are microplastics? As one might guess, microplastics are microscopic plastics that range up to 5 millimeters in size by definition. There are 2 different types. Primary, which are manufactured in that size range. Think those abrasive beads you have in facial cleaners or things really in the beauty and health industry. There's also secondary, which are weathered from large plastics called macroplastics. And this is from wave action that they get broken down from biggers into smaller and eventually microplastics. It could be sunlight, could be mechanical weathering, could be anything. But the most pressing issue is that they've been reported in every system on Earth, from the Arctic tundra to the atmosphere, believe it or not, to deserts and everywhere in between. So why should we be worried? As I said, they are persistent in aquatic environments, and my friend Mr. Plankton here, you can see he's eaten some microplastics because they mimic food. And not only does Mr. Plankton not get calories, but there's also a chance that chemicals can stick to these microplastics, and when he eats them, it's harmful to the plankton and everything else that eats it, and eventually we eat whatever Mr. Plankton eats, right? So that's a process called biomagnification, and that is not very good for anybody involved. So the research of microplastics, the study of plastic pollution in general is relatively new, dating back to only the 1970s. But microplastics is even more infant, with the term microplastics being coined in 2004 in a research paper. Most research has been done in marine environments, think beaches, coral reefs, and coasts, and things of that nature. And there's little research regarding microplastics in terrestrial fluvial environments, fluvial meaning river, like the Pearl River, that is just right there. So why the Pearl River? As I said, the contamination has been properly documented on Gulf Coast beaches, and they're just not common. To my knowledge, this is the first known study in Mississippi, maybe even the Gulf Coast. I'm not entirely sure about that, but for sure, Mississippi. So the objective here is to document the microplastic loading in the sediments of the Pearl River, and through this we can obtain information related to plastic loading through the urbanized areas of this metropolitan area. And then we can shed light on the effectiveness of wastewater treatment plants at removing microplastics that go into fluvial terrestrial environments. So the area I studied was a 35-kilometer portion of the Pearl River between the Ross Barnett Spillway and a little south of the Savannah Street wastewater treatment plant. I dubbed this the Metropolitan Watershed. As you can see, the watershed is the blue lines. So the Metropolitan Watershed is comprised of 11 urban watersheds that all have creeks that are part of it, and they flow into the Pearl River. For comparison, the Metropolitan Watershed, seen in dark gray, has a population density of 369 people per square kilometer versus the Ross Barnett Reservoir Watershed, which is much larger, but only has a population density of 22 people per square kilometer. And this is, of course, to focus on the loading of microplastics and the effect of a large metropolitan area in urban areas. So what was sampled? I sampled point bars amongst the Pearl River. Point bars are just these sandbars that happen when a river meanders and sand is deposited. Through floods and things like that, plastic has a high chance of being deposited in these point bars, so that is why I chose to sample these point bars. Here is each black dot indicates a sample point. There's approximately 57 samples were taken. There's approximately 1.8 samples per kilometer, and that meander belt right there in the middle of sorts, that has no samples because that's where the river was channelized by government entities to prevent erosion and protect the infrastructure of the city of Jackson. So Dr. Galicki and I loaded our kayaks on his Ford Explorer, and then we went down to the Pearl River and dragged them all the way down the embankment and took off. So this is the Pearl River. On any given day, it could be it's a gorgeous river, very scenic. On the left middle there, that is a great heron, and it's hard to see, but on the right, that is a bald eagle sitting on top of a tree, so teeming with wildlife as well. And me and Dr. Galicki made a startling discovery of Millsaps Falls right here on the right, and we call that Major Arch right here on the left. So our sampling method was tried and true, never done anybody wrong, just paddled up beside a point bar, took our paddle, scooped some sand into a plastic bag, done and dusted, next one, right? So once we lugged all of those samples up the embankment, again, this was not easy, I set them to dry on styrofoam plates on baking sheets, first to air dry, and then afterwards I transferred them to mason jars and to dry in the oven overnight to ensure that there was absolutely no moisture in the samples so that they would not be interfered with the solution, which I will talk about later. The next step was a particle size analysis. We used a gravimetric particle size analysis in which this machine on the right here, that's a Rotap shift shaker, and these mesh numbers, 10, 18, 35, 60, 120, and 230 correspond with particle size boundaries. That is an industry standard. And yeah, we put them in the machine and it shook until the particles could not go any further, indicating that particle size. So the density separation. I used a high-density sodium iodide solution, which ranges from about 1.52 grams per cubic centimeter to 1.55 grams per cubic centimeter. Quartz sand is 2.65 grams per cubic centimeter, meaning it's more dense and it would sink in the solution. However, plastic is, as you can see on the right, this is a chart of all of the 6 most common plastics used in the world, and the highest density is 1.45 grams per cubic centimeter. So in theory, all of the plastics should float because they are less dense than the solution. To ensure that my method of density separation would work, I purposefully contaminated a clean sample of Mason sand with microplastics and processed it with the processor, which I'm about to get to. And it turns out it worked. It was not quantitative, that test, but a substantial amount was recovered to encourage me to go ahead with the experiment. From then, to make sure what I was looking at was microplastics, I used a soldering iron, which is just a hot tip, and I used it to melt the plastics. If it melted, it was plastic. If it didn't, it was not plastic. So this is the microdensity separator, and when I tried to explain this to myself, I found it hard, so I brought the one that I use, and I'm going to use it as an example. So imagine, if you will, that there's a magnetic hot plate right here, and there's a magnetic stir. Once you will put the solution up to this black line, I don't know if you can see that, and then you put the sediment in, and you stir it for 10 minutes. And then once that 10 minutes is over, you let the sediments settle for 10 minutes, and once that is over, you take it and you slide it across like this. So in theory, all of the plastics should be up here caught in this portion of liquid of the solution, and all the sediments should be down here in this portion of liquid. So after this, I took this top half right here with the theory of all the plastics, and I put it in a vacuum filtration system with a glass microfiber filter with openings of 1 micron, which is incredibly small. Nothing, especially not plastic, we'll get through that. So I poured that into a funnel, I vacuum filtered it through the funnel onto the slide, and then I used a Nalgene bottle on the right here to ensure that any plastics stuck to the side of the funnel would not be lost. They would go onto the filter, and then I vacuum filtered that as well. And here is just an example of a sample. This is a glass microfiber slide that I just mentioned, and I stuck that under a microscope and looked for microplastics. So one problem I was running into early on was I was looking at these slides and I said, hey, wow, that's a lot of microplastics. But once I used a soldering iron to melt them, they wouldn't melt. So I determined that there were filamentous algae, and to reduce the chance of me seeing filamentous algae, I used hydrogen peroxide to dissolve all of the algae. And on the right is microplastics, on the left is filamentous algae. As you can see, they're pretty easy to confuse, but once I incorporated the hydrogen peroxide into my processing, it got significantly easier. And these are examples of microplastics under the microscope. On the left is a bundle of fibers that seem to be tangled, and on the right, I'm 90% sure that is a plastic fragment of a plastic bag from Walmart or something like that. One thing that was a major breakthrough in this study was the use of different wavelengths of light to identify microplastics. As you can see, these are the same photo, but once I learned of the plastic chemical property that it fluoresces under black light, I looked at all the samples under black light, because originally I wasn't seeing that many microplastics even after I dissolved it in hydrogen peroxide. So as you can see, they are the same picture, but it's quite frankly and quite literally night and day between these two pictures. So the use of black light helped me identify much more microplastics. And now onto the results. The grain size analysis, as you can see, the overwhelming majority, 60%, were 0.25 millimeters in size, which is classified as a very fine-grained sand. So yes, and then the uniformity coefficient, which is how well something is sorted, how similar the particles in size are to each other, is 2.5, and we used a graphical analysis to determine that. So in the end, this is a fine-grained, well-sorted sand. Now on to the microplastic loading. 652 microplastic particles were identified in 57 samples. The majority of them were fibers, 96% to be exact. Very little spheres and fragments. The average loading of one given sample amongst the 57 was 363 particles per kilogram. That's how many particles you'd find in a kilogram of sediment if you were to just arbitrarily pick up a kilogram of sediment. That's how many you'd find. And they range from 0 to almost 5,000 particles per kilogram. One water sample was taken at the outflow of the Savannah Street wastewater treatment plant, and that came out to 73,000 particles per cubic meter. So going back to the metropolitan watershed, this is each red cross represents the loading of the individual loading of each sample, and then the creek names. There seems to be no correlation between microplastic loading and the confluence of the creek with the Pearl River. And here is just the stretch of river that I sampled, with each black dot being a sample, and the loadings per kilogram of next to each black dot. And then here it is continued. And this blue area right here, that is the Savannah Street wastewater treatment plant. So the main point of this was to compare my study with studies of that around the world who have conducted similar experiments. And to put it frankly, they vary all over the place. So in Europe, for example, in Central Europe, we are considerably less polluted than Central Europe, and in England for 40,000 particles per kilogram compared to my 363. That is quite the crazy discrepancy. But however, in Italy and the Tibetan Plateau, which is, might I say, less densely populated and less industrialized, are significantly less, more concentrated, that's the word. So yeah. So there's every chance for me to see more microplastics. The further down the river I went, I saw a qualitative increase in floating macroplastics and debris. Every reason for microplastics to be created, so why didn't I see more in the sediment? Rivers like the Pearl River, they do have a residence time of about 12 to 20 days, so that means that from the headwaters all the way down to the Mississippi Sound and eventually out into the Gulf of Mexico, that water is only in the Pearl River for 12 to 20 days. And this corroborates the fact that high concentrations are reported on the Gulf Coast where Pearl River comes out. There are no high concentrations close to the spillway, which means the reservoir is acting as a possible sink. And the Pearl River is not a turbulent river. There's no wave action that can consistently break up all of the plastic and turn it into smaller plastics. Other sources of microplastics might be the Savannah Street wastewater treatment plant. However, one water sample taken is not enough. So in future studies, which I will get to in a minute, we will have to take more water samples. So in conclusion, it is a fine-grained, well-sorted sand with an average of 363 particles per kilogram, ranging from 0 to almost 5,000 particles per kilogram. Results vary around the world with land use, population density, and levels of industry in any given area being the main factors. The metropolitan watershed has relatively low population density and a low level of industrial activity, and I believe the wastewater treatment plant does sequester some microplastics. However, like I said, more samples need to be taken in order to determine a more quantifiable rate. More water samples, this is what to do next time. More water samples both past the Savannah Street wastewater treatment plant and throughout the study, just in general. One thing we could do is partition point bars into square meters and then get the concentration of microplastics per square meter in order to determine both the particles per kilogram and the particles per meter squared. There's also, you can use different wavelengths of light, not only to identify more plastics, but to determine what type of plastics are most abundant. And this research is still new, but it fills a gap in microplastic research in the Gulf Coast. And that's the last slide. But I would like to acknowledge my committee. First of all, Dr. Galicki, thank you so much. Without you, I'd be stuck up a creek, both literally and figuratively. Dr. Harris, without your kayak, I'd probably still be paddling up the Pearl River. Dr. Hendrickson, thank you for being my third reader. My friends and family, obviously, brothers of Sigma Alpha Epsilon, Phi Alpha. That's it.
All right, does anybody have any questions for Willie? Abby.
So based on that chart of like the amount of pollution in like rivers across the world, if you're able to do this, how can you predict the like pollution habits of people in Jackson based on your findings?
It's really hard to predict pollution habits in the Pearl River, specifically because there was no correlation between the creek confluences where most of the people live in those 11 urban waterways. There was no correlation between those, so I'd say that's more of a sociological question, I think, but there's no way to quantify, considering also the Pearl River's constant flushing.
Is there any fear of contamination with the styrofoam plates that you were using, or just not statistically significant?
That is an excellent question. I kept an eye out specifically for styrofoam plate fragments in every sample I analyzed, and I did not see any. That's the same with the plastic bags that I put the samples in and the paddle that I used to scoop the samples in. So that's a great question.
Sample size is just random throughout your area that you were?
No, so all of the sample sizes that fit in this was 30, approximately 30 grams went into each one. But I managed initially to dry out, I set 500 grams aside, because that would leave me enough both to do that and to do the particle size analysis.
Did you notice any differences as far as like where you took the sample and as far as the concentration of microplastics in that sample versus maybe somewhere that might have less next to it?
No, there was
or was it just randomly?
Yeah, just yes.
Let's thank Willie one more time. I got to say, our students do much better than senior researchers at sticking to their allotted time. So we're right on, perfectly on schedule. Just take one more minute. Oh, sorry. Let's see, there we go. So this is our very last presentation of the 2024 Honors Conference. It's an honors thesis in anthropology, and the title is Inca Imperial Law, A Comparative Power. And give a warm welcome to Bernard Booth.
We're at the finale, y'all. We're getting close. I first of all just wanted to thank my honors committee, Dr. Belil and Dr. Yates specifically. They were great helps in poking poking at all the flaws in my argumentation, and that's awesome, even if it does hurt. I'd like to thank my parents for supporting me and sometimes dragging me along when I thought I really just do not want to do this. And I'd like to thank my fraternity brothers for being here and the rest of my friends as well. So thank all of you, and let's get the show on the road. So my interest in the Inca Empire really got started when I went to Peru, which is where they're from. And so this is me and some other people in the crowd. And in the back, you can see that site. That is Machu Picchu, one of the most famous sites in the world. It was an Inca imperial estate. And overall, just a pretty famous place to be. And I was looking at all that, and I was wondering, how are they doing this? Because you don't just manage to drag all these stones up that hill. As you can see, it's pretty vertical, so you've got to work on it. So first, I'm just going to give you all an outline of just what I'm going to do. I'm going to explain why I'm comparing the empires I'm comparing. I'm going to give you the basics of these empires. Then I'm going to give you just some really cool facts about the Inca. Then I'm going to do a compare and contrast just to make life easier. So first off, I decided to compare the Inca and Athenian empires because they are so different. On the scale of what empires can be, they're almost complete opposites. They were thousands of years apart, thousands of kilometers apart. There was no way for them to have ever met each other at any point in history. So they are, if you're trying to figure out what an empire can do or can be, about as close as you can get. First, the Athenian Empire, 454 to 404 BC, 50 years, not very long. Roughly 2 million people, not very big. They were competing with the Peloponnesian League, which is in red here, and they were local competitors with each other. They spent a lot of time fighting, and you can see that giant orange blob, that's the Persian Empire. They stretched all the way from Turkey to India. They were massive, and they were a superpower that these two little empires existed on the periphery of. Important for the context of my thesis, looking at law, they had something like a federated legal system. They let each Greek city-state run their own affairs for the most part, but if they decided something was important enough, they would interfere with that legal process. And that just kind of stands in general for what they did, but largely they relied on each city-state to run themselves, kind of like our state and federal system. Very similar idea there. Moving on to the Inca Empire. They lasted from around the early 15th century to 1532 AD with the Spanish arrival. They were roughly 12 million people, so much, much larger. They did not have a system of writing. And Inca is the name of the dominant ethnic group, and the empire is named after them. But inside of that empire, there are tons of different ethnic groups that spoke different languages. They wore different clothes. They were distinct peoples that had their own ways of doing things. And this is modern-day Colombia, Ecuador, Peru, Bolivia, Chile, and Argentina. Interestingly enough, there is an incredible degree of topographical variation. You go all the way from the coastline, up 15,000 feet, and back down to the jungle floor in this empire. So again, I was looking at those stones thinking, how are you doing that? How are you dragging these from such wide variety of locations? How are you controlling all of those people? And this brings me to Quipu. This is a huge portion of how they were doing it. Like I said, they didn't have a system of writing. What they did have were Quipu, which is a mnemonic rope device in which you would knot up this rope, and it would tell you information. So for instance, let's just say 1 knot equals 1 bunch of potatoes, or whatever system you want to use. We don't really understand the inner workings of it, but we do know that this is how they recorded information. There were officials whose entire job was to do these things. And importantly, just to give you a little bit more context, the Inca had a command economy, meaning that they controlled pretty much any interaction. So whenever a potato was taken out of the ground, they recorded that. Whenever it was put into a warehouse, they recorded that. Whenever it was put back into the ground, they recorded that. And they relied on feasting pretty heavily. It was something that they did. There were religious festivals pretty much once every 10 to 14 days where there was food provided. So there were always really large parties where you were expected to eat and drink, and the government was paying for all of that. And that will come up later. But this leads me specifically to where law comes into these things, and it's kind of a long slide. So it is all predictable and accountable reactions by the imperial apparatus to the actions of those subject to it. They are the rules, written and unwritten, that would prompt the imperial state to intervene in cases which are not inter-community conflict. If a dispute is solved amongst locals, it is outside the purview of the study. That is the most science I'm going to get on you, don't worry. What it really breaks down to is, if you do something that the Inca don't like, and they whack you for it, you broke a law. If you do something that the Athenians don't like, and they whack you for it, you broke a law. Kind of the moral of the story. And like I said, it has to be unwritten rules because I'm dealing with an empire that has no system of writing. But there's clearly some way of communicating the expectations of the boss to the subject. So moving forward, I'm really going to focus on the Inca in this, but so I'm going to again go over those key findings. First up, Andean ideas about land ownership. So the Inca Empire had this notion that all land belonged to the crown. It belonged to the king. Other people were allowed to use it, but it was the king's land. So that's the first big concept. The second big concept that we need here is that land ownership is expressed by feeding the people that work that land. Now you remember that feasting that I talked about? That's important. This is how they expressed land ownership. They had to do this feasting. And in fact, this is important because their entire system of taxation is based around this concept that the Inca cannot simply take stuff from people, because if they simply took food, then it would be acknowledging that they owned that land and that the Inca really didn't have a claim to be there. So instead, they had to frame everything as a form of labor taxation. So this labor taxation is how they did everything, and it's how they built those giant mega projects, it's how they hauled stones. They had roads, they had inns, they had warehouses, and they had to frame all of this as a form of labor taxation, not taxation in kind, not currency. So that's a really important behavior to look at. And in fact, you see this giant lake in the middle here? That is Lake Titicaca. It is one of the highest navigable bodies of water in the world. And there are fisher communities that have been there for thousands of years. But when the Inca king shows up and he says, actually, I own all the land and I own all the rivers and I own all the lakes. I own this lake. I already do, and I have since like time immemorial. You guys just didn't know about it. So he was claiming that he was giving people a portion of their cash back, and he was only taking stuff from what he already owned. Obviously, if you look at this, it does not make sense. But legally, this is what they had to do to justify their empire. Now, moving on to some of the similarities and differences. Similarities. The biggest one is that there are a ton of legal protections for the ethnic in-groups and their allies. For the Athenians, what this looks like is that the Athenians gave themselves special rights and to their proxenoi or ambassadors within their own communities. And these legal rights took a couple of different forms, and same for the Inca and their allies called Inca by privilege. The first big one is that important positions were reserved for the in-group. So for instance, within the Inca legal system, there were like 11 levels of authority. The top 3 are all reserved for Inca, and then the next 2 are all reserved for their specific ethnic allies that are given special titles and legal designations. And for the Athenians, the archons, so the people that could judge trials, the people that could be treasurers, and the people that could have militaries, all had to be Athenian. And then proxenoi, or ambassadors, were given special legal protections in and of themselves. What are these legal protections, you're asking? Well, in both cases, the only person that could try an Inca and the only person that could try an Athenian were an Inca or an Athenian respectively. In other words, any form of murder trial could not be tried by a subject of the empire. They had to be tried by the ethnic in-group. And this holds true for their allies as well. So any kind of important trial was moved to either an imperial center or to a capital, and it could only be dealt with by an Inca or an Athenian. So they're interfering with the legal processes of their subjects to maintain control and to make sure that their allies or themselves don't get legally persecuted or punished whenever they want them to be safe. Well, what does this look like kind of in a statistical manner? What this looks like is the top 2 to 8% of the population have incredible legal rights compared to the bottom 92 to 98%. What this means is they have special murder trials. They normally can't be issued the death penalty, or if they can, it's only in incredibly specific circumstances. The trials are involving fraud or other forms of behavior can only be tried by them as well. And that's kind of the biggest similarity that I found. They're using legal protectionism to make sure that they can't be hurt by their own subjects under a supposedly fair framework. But there are some rather large differences. For instance, these empires have different ideas about what control looks like and the degree of control. The Athenians, like I said, used a federated system. They relied on each city-state to maintain their own affairs, and they had their own systems and ways of doing things that the Athenians largely left alone. While the Inca, if there was a village of 100 people, there was somebody that had an Inca title and position within that community. They interfered with it. Again, like I said, they had a command economy. So if you put something in the ground and you took it out, the Inca state was recording that. They were monitoring your behavior at that level. And the degree of centralization. So Athenian state officials had relatively little authority. What I mean by that is you had one guy that could run a trial. You had another guy that could be the treasurer, and you had another guy that could run the military. These were separate titles. Versus the Inca, it didn't work like that. The person that's in charge of 100 households as judge, jury, executioner, administrator, and treasurer. They ran all of it. There's no breaking up of duties like that. And so that's what I mean by degree of centralization, which is a huge difference. But, interestingly enough, there's an incredible degree of the difference in investment or overhead that you're seeing in these two empires. Running the Athenian state required maybe 2,000 officials max, really not a large number. Versus the Inca state required tens of thousands of officials. And a relatively minor imperial center up close to Lima, there were 2,000 officials, and that's like more than the entire Athenian empire combined. But again, the reason why is because they had such fine control over the behavior of their subjects, but that also required an incredible degree of investment and time. So to wrap this all up, and for me to just draw it all together, these two empires were divided by a huge gap in space and time. Thousands of years, thousands of kilometers. These people would have literally never spoken to each other ever. But there's still some points to be made about them being similar. They are very interested in controlling trials and who can hold office. So they're making sure that themselves and their allies are protected, that they're the ones that get the best jobs, and that they can't be hurt by their subjects. But there are some rather large differences in the scale of these empires, the degree of control necessary, their ideas around centralization and who should have power and how much. And so this kind of led me to a conclusion, which was that empires are always unique phenomena, but they are fundamentally one ethnic group dominating another, and this leads to unique legal phenomena, and where the specifics of these laws can change, their intended effect is often the same. Thank you all, and any questions?
Any questions for Bernard?
So you mentioned that the Inca had a lot more government officials than the Athenians. Would you say that the payoff of having more government officials and greater control over those areas paid off for the amount of investment?
Well, so like I land economy, which leads back into that feasting, which leads into my point about land ownership. They literally had to invest this much in order to be perceived as legitimate. They had to run those warehouses, they had to have those feasts, and the only way that you can do that is by having this incredibly fine degree of control over the subject population, because again, they can't just accept payment outright. They have to frame it as them feeding the people working on their land. And the only way that you're going to manage to feed 12 million people is by having an insane degree of control over what those people are doing so that you can make them produce stuff for you. So it paid off, but there's some very specific reasons why they're doing what they're doing. Mm-hmm.
Eric?
What primary or secondary sources impressed themselves on you?
So when dealing with the Inca, like I said, they didn't have written sources. And so the only people that we have that interacted with the Inca Empire, like as an empire, were conquistadores, who were very specifically interested in how do I murder the people in charge and then tell the other people, actually, I'm in charge now. So their perception of, Peru, the Andes in general, is very skewed. And we only get these kind of retrospectives on the empire 40, 50, 60, 70, 80 years later. And so having to piece together those like first-hand accounts with those accounts that are a little bit more removed was a bit of a challenge. And so the thing that impressed me was how much disagreement I found there, right? Like in just 80 years, people's perception of what had happened changed so rapidly. And then of course there's the secondary scholarship where everyone has their opinion on what happened.
Laura, did you also have a question?
Yeah, so then has anybody else compared the two specific examples that you compared? And then, or was that your idea to compare these two? And so what made you, I mean, you kind of alluded to this, but then what makes that an important step in original scholarship to compare those two?
Right, no, as far as I can tell, no one's compared these two. What I found is there's kind of this weird gap where people want to view an empire as its own phenomena and not really put it in direct comparison with another one. Or I found scholars that are really interested in viewing empires writ large, but they're not necessarily, there's not that middle ground, right? Where someone's putting 2 to 5 empires in comparison with each other to try and see what you can draw out from that. And so there's a Dr. Sinopoli who kind of built this theory of empire and what they tend to do, but she doesn't like have these granular approaches where she's comparing just two. And why I compared these two was very specifically, like I said, they have no dialogue with each other. The last time that someone who could have theoretically been an ancestor of the Inca Empire theoretically talked to a Greek person was like at least 10,000 years before either one of these empires got started. So they had such different origins that to end up at similar enough points that they're able to be compared means that like there's a unique social phenomenon happening there that like humans tend to or are able to do, you know?
Thank you.
Last questions for Bernard? Let's thank Bernard again. So we're now going to take about a 15-minute break. I encourage everybody to get more food and drinks. If anything's running low out there, I have more drinks in the fridge. We'll congregate back here in about 15 minutes, and we'll present some awards given by our chapter of Phi Beta Kappa. So see everybody soon.
Okay, so I think, oh, now it was too loud, sorry. Okay, now we will gather back here for just a moment. What a wonderful afternoon it has been to see and hear these wonderful presentations from different parts of the college. So it's always great for people, you know, in the sciences to hear what's going on in arts and humanities and vice versa. So I am a member of our Phi Beta Kappa chapter here at Millsaps. It's the Alpha chapter of Mississippi, the first one, the best one, right? Anyway, but it's wonderful that we love to, you know, see this great research being done and the wonderful work that these students do. So today we have a presentation for a prize for the best honors project presentation. So a little cash prize in here. And so the prize this year goes to Willie Ponte.